This is a spinal cord sketch. I'm trying to draw the gray matter that's in the spinal cord. Let's do it like this. I'll try and do this mirror image the best I can. Let's start there. Let's start labeling this spinal cord sketch that we have. I've tried to put these gray horns, and I don't have a gray color, so I've just drawn them in a brown color. But we're going to have what we call the anterior gray horn. This would be our lateral gray horn. And our posterior gray horn. You'll see in some slides and some textbooks the word anterior and ventral are used. This could be ventral gray horn or anterior gray horn. Just like dorsal gray horn or posterior gray horn are interchangeable. And maybe one clue you might be thinking, how do you know which one's posterior and which one is anterior? And a clue are these differences that I've drawn into this diagram right in the middle. On the anterior side of the spinal cord, there's this deeper separation between the two halves. It's a deeper separation we call the fissure. This is the anterior median fissure. Anterior median fissure, and then you can see around the posterior or dorsal side, there's just a little shallow depression that we call the sulcus. So that's our posterior median sulcus. Okay, so we've got the gray horns labeled, and you can see the gray horns are also over here. I'm going to put some different things on those gray horns, so I'm just going to label them on this side and just, realize those are named the same thing, just on the opposite side. And here's what I'm going to try and add to this. Let's maybe put some type of key up on the board. Here in the middle, I'm going to use these different colors to represent parts of the central nervous system that we talked about before. When we were talking about functional classes of neurons, we were talking about sensory neurons, integration neurons, and motor neurons. I'm going to put what we will call somatic sensory in this darker blue color. And in a lighter blue color, I'll put visceral sensory. What we knew about both sensory neurons is that they were sending signals from the peripheral nervous system, from outside of the brain and spinal cord, always towards the brain and spinal cord. We called this the afferent pathway, going towards the brain and spinal cord. Then within the brain and spinal cord, there were the integration neurons, sometimes also called association neurons. And then in either a red or an orange color, I'll put the motor division. We've got the somatic motor. You may remember somatic motor. This is the branch that goes to skeletal muscles. So we're in voluntary control of this branch, but the other branch of the motor division we were not in control of. That's the autonomic branch. So the autonomic motor division is going to go to the muscle groups that we don't voluntarily control, like cardiac muscle and smooth muscle. And you can see where we're going in Unit 5. We're going to talk about this autonomic nervous system and the two settings, that sympathetic or parasympathetic setting. We've got our functional classes of neurons. Let's try and map where those make their way into and out of the spinal cord. Because it's interesting, if if I were to expand this a little bit, let me try this. Okay, I'm happy with that. I'm going to put one identical to the other side that we can also draw, that we maybe don't have to label. Okay, we got mirror images of these things. And notice that on the dorsal side, or this posterior side, there's this bump that I'm drawing in there. Let's label those things. In fact, I'll just use this side over here to try and continue my labels. These little connecting structures that we've drawn connecting, really touching the spinal cord. The one on the anterior side, we're going to call this the ventral root. And the one along the posterior side, we're going to call the dorsal root. Notice that the dorsal root has this bump attached to it that we're going to call the dorsal root ganglion. This dorsal root ganglion, we've used that term ganglion before. We said it. Does anybody remember what a ganglion was before I give it away? It's a collection of something, that's right. It's a collection of cell bodies of neurons outside of the brain and spinal cord. So we're right next to the spinal cord, and these ganglia are gonna house collections of cell bodies of neurons. Let's see, the only other thing that I want to label on this side is this part right here that we're going to call the spinal nerve. You're just in time because I've just got the sketch of the spinal cord up and we're kind of labeling the parts. And I've left a clean side over here that we can try and draw some of the neurons into. And I've tried to color coordinate these neurons that I'm going to draw. Integration neurons, just to kind of hammer this point home, we talked about last time, integration neurons are always within the brain and spinal cord, and they never are found outside in the peripheral nervous system. Before I draw interneurons in there, I'm going to use these two different blue colors to represent the pathway that sensory neurons take. We know all sensory neurons, whether it's somatic sensory or visceral sensory, are going to be sending signals back towards the brain and spinal cord. They're going to make their way through the spinal nerve, and then they've got a fork in the road here. Sensory neurons are never going to take the anterior route. They always make their way, sending signals towards the brain and spinal cord through this dorsal root. And then right here in that dorsal root ganglion, I'm going to put a stalk and a cell body off to one side. And then we're going to put the axon portion, making its way right there to this posterior gray horn. This, without crowding this too much, is our somatic sensory. Just for simplicity's sake, I'm going to put the little dendritic end of the neuron out here. We've got the cell body off to one side. Again, cell bodies are located in the ganglions if they're outside of the brain and spinal cord. And then we got the axon end kind of terminating right there at that posterior gray horn. Structurally, what type of neuron did we draw? Well, so sensory would be the function. Yeah, it's a unipolar neuron. So we've got this one long dendrite. Sensory neurons always make their way in through this dorsal root. We would see the collection of cell bodies there in the ganglion. And this light blue one is a visceral sensory. So it's, if you have a stomach ache, we're going to see these go just a little bit further into that gray horn. Both sensory neurons, whether it's somatic sensory or visceral sensory, they're always going to send signals through this dorsal root Their cell bodies, again, located in that dorsal root ganglion, and their axons are going to terminate on that posterior gray horn. The integration neurons are going to be the ones that pick up that signal. So I'm going to draw a few of these integration neurons in here. Maybe I can label that somewhere in here. So we've got our integration neurons within the brain and spinal cord. They're picking up signals from the sensory neurons, and then they're conveying that information out to the motor division that I'm going to draw either in a red or an orange color. We're going to see that these are motor neurons. All integration neurons and motor neurons are going to be multipolar neurons. So let me draw multiple dendrites attached to those cell bodies. And I'll attempt to draw another multipolar neuron. If we're thinking about these somatic motor neurons, we're going to see that they have their cell bodies located in this anterior gray horn. When we were talking about gray matter, we said it was collections of dendrites and cell bodies and unmyelinated axons. So the cell body and the dendrites are in this anterior gray horn, and then the axon is going to make its way out this ventral root. Then as the ventral root merges with the dorsal root, that's when we call it a spinal nerve. A spinal nerve is a two-way street. That's one of the things I'm trying to draw here. Somatic sensory, they have their cell bodies in this anterior gray horn, and they're always sending signals away from the brain and spinal cord out this ventral root. You can say the same thing for the autonomic motor neurons. Their cell bodies are going to be located closer to this lateral gray horn. But again, their axons are going to make their way out this ventral root. And eventually, where both ventral root and dorsal root come together, this two-way street is what we call a spinal nerve. Spinal nerves, you can think of them as mixed because they're both sensory and motor. Whereas the roots themselves, like a ventral root or a dorsal root, these are not mixed. These are either completely sensory or a complete motor division. All right, so kind of just in time, I just have this sketch of the spinal cord up. We've got the gray horns, we've got the anterior or the ventral root and posterior root, and I'm trying to draw the neuron locations as they go through these ventral and dorsal roots. So if you had like some kind of freak accident, this happens. You know, I'm always driving down the road, I guess, and I'll see an 18-wheeler that has something weird hanging off the back, and I'm always worried that it's going to flip off of there and do some kind of weird stabbing me through the windshield thing. But you have these freak accidents that happen. Imagine if somebody had one of these types of accidents where their ventral roots were just severed and cut off. Right? Nothing happens. Their dorsal roots are fine, but their ventral roots are severed and cut off. I don't want to damage my diagram, but you can imagine if somebody's dorsal roots are fine, but their ventral roots are cut off, then they're able to sense pain. Their sensory neurons are working fine. They can pick up sensory information. However, when they go to try and control a muscle, the motor division is cut off. So this would be somebody that can feel pain but cannot move anything around. So paralyzed individuals that can feel pain but can't control muscle movement. If it was the opposite, if you had some type of damage to the dorsal root, but your anterior or ventral roots were fine, then you would have trouble feeling pain. You'd be able to move muscles around. Yes?
So the spinal nerve is when the autonomic and somatic motors mix?
So when you said autonomic and somatic, both of those are motor division. So both of those would be ventral root. Mm-hmm. That's right. But when you're saying mixed, that means motor and sensory division. We don't see the two-way street until we get to the spinal nerve. Remember, and I don't want to crowd the drawing too much, but sensory neurons are always sending signals in this direction, back towards the brain and spinal cord. And then the motor division is always sending signals away from the brain and spinal cord. And then whenever we get to this spinal nerve, we've got two-way traffic. We got sensory and motor signals going two different directions. But prior to that, we had these one-way street situations.
Okay, so it's when they all mix, the sensory and the motor ones
Mixed means sensory and motor. That's right. You couldn't say just sensory or just motor. And we'll do this, just a heads up, when we get into the cranial nerves, the peripheral nervous system, we'll list cranial nerves as either sensory, motor, or both sensory and motor. They can do. So it's interesting to think that when we get close to the spinal cord, these functional neurons go in different directions. It's all sensory in the dorsal side, and it's all motor on the anterior or ventral side. There are these little spaces that I have just left in kind of a white color. In fact, I'll come over here because it might be a little bit easier. I'm going to put one of these motor neurons, cell body and dendrite, in this anterior gray horn. We'll put their axon portion making their way all the way out to that spinal nerve. Let's put a sensory neuron making its way right there, and then I'll draw one integration neuron in between. Now that I've kind of drawn the rest of these neurons, it's really the fibers of those neurons that are going to separate these white areas into what are called columns. There is this lateral white column. It's made out of white matter, so you're going to see insulated axon portions of neurons. This, just behind these dorsal fibers, just maybe label it over here, this is our posterior or dorsal white column. And then anterior white column is right here. It's on this side of the root. So anterior white column, lateral white column, and posterior white column. There are only two other things, I guess, that I want to squeeze in here. And I'll try and make one of them right here. You've got these little patches of white matter. There's one on the anterior side and the posterior side. I believe your handout in class just wants you to know this area of white tissue that basically connects the two halves of this spinal cord as the white commissure. Technically, this is the posterior white commissure because there's an anterior white commissure right here. I'll just say it. Your lab handout doesn't distinguish between posterior white commissure and this anterior white commissure. And then this area, I guess I'll just use this space. Well, I'll try and keep it over here. This is too difficult. This little space of gray tissue that connects both halves of the gray horns is our gray commissure. And the only other thing to label is that dot right in the center. That's the central canal. That central canal is going to be filled with cerebral spinal fluid. It goes down the center of that central canal, and then we said it also surrounds the outside of the spinal cord. What I haven't drawn around the outside of the spinal cord are all of those protective meningeal layers. We can see the spinal nerve that is where the two roots meet. There's our ventral root and our dorsal root, and it's always the dorsal root that has that bump on it, the dorsal root ganglion. So if you were just walking down the street and saw somebody's spinal cord laying on the ground, you wouldn't need anything else other than those roots of the spinal cord to be able to tell what was the anterior side or the posterior side. I feel like that was the new stuff that we didn't get to last time, because right at the end of last lecture, maybe it was in lab, I was trying to point out these protective layers that wrap around the brain and spinal cord. And we were pointing out on the models this outermost protective layer around the spinal cord that we called the Dura Mater. Then underneath that Dura Mater, there was this kind of spidery-like layer that have all these fibry projections, which is where it gets its name. Arachnoid Mater means spidery layer. And then underneath that arachnoid mater, as we make our way closer to the spinal cord itself, you can see there's a layer of connective tissue that touches the surface of the spinal cord. That's this pia mater. Again, a brain formation or a brain system are just going to be multiple parts of the brain that are working together for a common theme or purpose. In the case of the limbic system, you can think of the limbic system as the emotional control center of the brain. That's one way it's described in the textbooks, emotional control center. But another way of thinking about this limbic system is I think of it as the bouncer of information. It's like one of those bouncers that determines if you're going to get into the club or not. And what it's if it deems the information important enough, then it gets into the brain. It moves into short-term memory. Once it's into short-term memory, you're going to have to mess with that information to transfer it into long-term memory. But again, if the bouncer of information doesn't think that that is important enough, then it never makes it into short-term memory. There are other parts. So there's multiple parts to this limbic system. You can see one part is that olfactory bulb. We saw the olfactory bulb last time, and I'm looking for these half-head models because dangling from that olfactory bulb is where we found those olfactory neurons. They drop down into the superior part of that nasal cavity, and they're responsible for picking up smells. And smells are things that can cause emotional responses. We can see how it's all tied together to the same part of the brain. And again, I talked about one of my buddies the other day that used to play music and wasn't able to anymore. And he now works for bands that go on the road and put on shows, like big bands. And one of the things they've been doing, he was telling me they take these fans and they put in different smells. I guess it's just like a big Glade plug-in or something like that. But they blast these smells, in pleasant, I don't know what they smell like, into the crowd as they're playing different songs. And that they're trying to trigger different emotional responses. I don't know all the different sensory things they're trying to hit there. But we can see the olfactory bulb kind of tied into this emotional control center. So again, limbic system, think emotional control center has something to do with regulating whether or not information is important enough to move to short-term memory or never gets in at all. So we've said that. The other system that I wanted to introduce is called the reticular system or reticular formation. One of the main jobs of this reticular formation is regulating whether or not you are asleep or awake. And here's a picture. This purple area that we see is this reticular formation, this reticular activating system. And what we can really think of this diagram as showing us is it's a collection of interneurons that are all touching one another like dominoes. And if we can get one of those neurons to start sending signals, it's going to cause the other surrounding neurons to start to send signals. Notice how we've got, well, we can't see auditory impulses. They just labeled auditory impulses as these blue arrows that are making their way into the midbrain portion. We know, as a review last time, these auditory impulses are going to hit that bump right there, that inferior colliculus, which is part of the midbrain. And visual impulses are going to hit the superior colliculus right there. So we can see visual and auditory impulses go into the midbrain, which is where they're picked up. And like we said, if one neuron starts pushing on another neuron, it's going to wake them all up. So let me use this analogy. If auditory and visual impulses are touching this reticular activating system, and we want to go to sleep, one of the things that typically help people go to sleep is a quiet, dark room. So there are no impulses that trigger this reticular activating system. And in a quiet, dark room, it's easy to fall asleep. And the opposite happens. Think about whenever you're waking up in the morning from a dark, quiet room. For me, it's usually, I'm never really lucky enough to sleep late enough. I remember a time in my life where I used to be able to sleep late enough where the sunlight coming in the window, when the room got bright enough, the signals coming in through the visual superior colliculus eventually got strong enough as the room got bright enough that, as these neurons start waking up the other surrounding neurons, this is also a good review. look at this reticular activating system, everything in purple. If we wake these neurons up, you've got some neurons right here next to the pons. We said the pons held those neurons that regulated respiratory rate. So we can increase respiratory rate by waking up these neurons in the pons. And we said there in this medulla oblongata, there was heart rate was regulated. So as the room starts to become bright in the morning, or how it is for me now, I usually have dogs that make noises in the morning that wake me up. And if there are these auditory impulses that really start to wake up the surrounding neurons, you can see as this reticular activating system starts to wake up, the pons starts to cause my breath to increase. You know, I'm starting to wake up, my heart rate starts to increase. And at a certain point, I have to get up and let the dogs out or whatever I have to do. But reticular formation, again, everything here in purple is regulated, or trying to help regulate whether you are going to be able to fall asleep or regulating whether you're waking up. And I used it as a review to remind us that the brainstem area here houses areas like this midbrain. Superior colliculus picks up visual impulses. Inferior colliculus picks up auditory impulses. The pons regulates respiratory rate. And this medulla oblongata down here regulates heart rate. It's also responsible for what we said, those vital reflexes like sneezing or coughing. So you can imagine if you're in the ER and somebody's having trouble sneezing or coughing, you can tell that maybe there's damage to that medulla. Okay, so two systems of the brain that we introduced, the limbic system and then that reticular formation or reticular system. The next slide just points out the two different stages of memory. I've talked about short-term memory and long-term memory before, and you can think of, I sometimes like to use this table up front, this single table, as a representation of short-term memory. Because like this table, in your short-term memory, you have only so much working space in short-term memory. It's limited to 7 to 8 pieces. So there's only so much I can fit on this table before things start to fall off. And once you have your short-term memory kind of loaded up, you're going to need to be able to do, there are certain tricks that you can do that converts this information from short-term memory into long-term memory. And here's a little graph that's just trying to show what I mean. We've got multiple sensory inputs that are coming in. And like we said, it's the limbic system, the bouncer of information that decides whether this input is good enough to make it into short-term memory or whether it's just lost immediately. Right, so this temporary storage buffer is our limbic system. Maybe a pleasing smell might get it past that and directly into short-term memory. So once we're into short-term memory, we will lose this information if it is not practiced. So again, if you don't use it, you lose it. I mentioned songs that we've had to learn from even last year, that if we don't play those songs over and over, I totally forget how to play those songs. They were in short-term memory for a while and now they're gone. But ways that we can trick our brain to take information from this short-term box and move it into the long-term memory. I talk about, well, for the band purposes, it's rehearsal. The more we can practice and the more I can play that song over and over, or the more you can fill out these blank handouts from memory over and over, that rehearsal transfers this information into long-term memory. So I definitely talk about that. I use my own personal family examples a lot about, we're lucky in here talking about anatomy and physiology. I mean, we all have a spinal cord, so we have this stuff. We can relate it to things that we either have dealt with personally or family members that have dealt with stuff. So making it feel important is another way to get things to move into long-term memory. You'll notice there is a side path here that takes sensory input and moves it directly into long-term memory. These are typically traumatic things that you see and immediately are burned into long-term memory and you'll never forget them. That's not always a good thing, but there are things, again, that are so extreme, you know, that they go directly into long-term memory. Okay, so those are kind of some of the basics of short-term memory versus long-term memory. And when we were talking about memory being adjusted at the very beginning of this unit, we said there were one of those types of cells that were responsible for changing those dendritic spines and those axon terminals. Does anybody remember which one of those cells it was? It also cleared out debris, any cell that didn't belong, any foreign substance, any pathogen that happened to make its way into the brain or spinal cord. It was the microglial cells. They act as part of the immune system that roam around that central nervous system and clean out pathogens, but they are also responsible for reshaping those neural connections. All right, so let's see where we go. Meninges, we just talked about those protective layers around the brain and spinal cord. They basically cover and protect that brain and spinal cord, the central nervous system. We're going to see underneath the arachnoid layer is where that cerebral spinal fluid is going to be found. And then some of these little expansions of this meningeal layer are going to partition the skull off. We've already introduced the Dura Mater, the Arachnoid Mater, and the Pia Mater. So here are some pictures of this. Again, the half head is the best place that you're going to see these two parts of the Dura Mater. There's the periosteal layer that we said is the part of the Dura Mater that touches the skull, touches the bone. And then there's another part of this Dura Mater that we call the meningeal layer, and they're kind of showing that in a more of a tan color, I guess. You can see the meningeal layer of the Dura Mater actually makes this way down in between those cerebral hemispheres. This is dropping down in that longitudinal fissure, separating both cerebral hemispheres. You have on your lecture handout, you'll have to know that layer of connective tissue that drops down into the longitudinal fissure. It runs between the two cerebral hemispheres. It's called the falx cerebri. So falx cerebri in this longitudinal fissure only.
And that's connective tissue, you said?
It's connective. It's part of this Dura Mater. That's right. It's the meningeal layer of that Dura Mater that has penetrated in between those two cerebral hemispheres. So you find it in that longitudinal fissure. From this same exact picture, if instead of thinking about the Dura Mater, if we switch, again, let's say we're still talking about the Dura Mater. In the skull, there's two layers of the Dura Mater. Like we said, in the spinal cord, there's just a single layer of Dura Mater. Here is another picture of the skull. We can see going through that longitudinal fissure is that patch of Dura Mater we called the falx cerebri. And then in between the two cerebellar hemispheres, there is a falx cerebelli. So it does the same thing as we see in the cerebrum. It runs between those two cerebellar hemispheres. And then I wish that, that looks like it's pointing to the blue part, but this tentorium cerebelli refers to this horizontal piece of connective tissue that it runs between the cerebellum down here and the two cerebral hemispheres. So this horizontal layer of connective tissue that runs between cerebrum and cerebellum is our tentorium cerebelli. Falx cerebri, falx cerebelli. We're just looking at these invaginations of the Dura Mater that run between the two cerebral hemispheres or the two cerebellar hemispheres, or separate cerebrum from cerebellum altogether. And again, you can see how tough that Dura Mater is along the outside. Back to this next picture, the same one that we were just looking at, but I wanted to point to the Arachnoid Mater this time. If we're thinking about that Arachnoid Mater, it's kind of easy to see because it's the layer that has those spidery-like appendages. So that Arachnoid Mater, we can see those little spidery-like appendages, and it's really within those spidery-like appendages. If we were walking through those spidery-like appendages, we would be in this subarachnoid space. It's the space between the Arachnoid Mater and this Pia Mater that we can see as kind of a tan color that directly touches the surface of the brain. I just want to point one other thing out right now. This arachnoid villi, you can see these are little projections that allow the fluid that would be moving through this subarachnoid space. We said that cerebral spinal fluid is what circulates within this cerebral spinal, let me say it again. Cerebral spinal fluid circulates in this subarachnoid space. And if we had a buildup of too much cerebral spinal fluid, how that excess cerebral spinal fluid escapes this layer is through these arachnoid villi. And it goes directly into this superior sagittal sinus. It's a big vein that runs down the center of the skull. That half head model that I kept showing, you can see the blue layer that represents this superior sagittal sinus. Picks up that excess cerebral spinal fluid. If we remember where that spinal fluid started, I'm going to jump to this picture. Cerebral spinal fluid began by leaking out of a capillary bed, so leaking out of blood vessels. And if we go back here, it goes right back into veins. It's returned to the circulatory system. So selective blood plasma is all this cerebral spinal fluid is. So now we've seen those protective layers around the brain. We said meningitis is just typically a bacterial infection of one of those meningeal layers, and that swelling puts pressure on neurons and causes problems. That cerebral spinal fluid, we said, was selective brain plasma, mostly water. There are some ions and some nutrients. It functions really to give the brain and spinal cord some buoyancy, like it's floating in a column of water. Think about the people that are rehabbing a damaged ankle or a knee joint or something like that. They typically put them in a swimming pool where they're not having to deal with all of their own weight, and they can rehab some of those damaged areas. Here's the same picture we were just looking at that shows the connection of these ventricles. There are these large lateral ventricles. We can see one lateral ventricle that has its choroid plexus. The fluid produced in this lateral ventricle eventually makes its way into the third ventricle through one of those interventricular foramen. This third ventricle has its own choroid plexus that it's also producing cerebral spinal fluid. We said the fluid from the third ventricle makes its way through this cerebral aqueduct and down into the fourth ventricle. That fourth ventricle is always between the cerebellum and the pons. So there's our fourth ventricle. It has its own choroid plexus. And as we drop below this fourth ventricle, that's where we are about to enter the spinal cord. And before we enter the spinal cord, there are these little exit opportunities for some of that fluid. These are the lateral and median apertures. And in our flow chart last time, we're showing that this cerebral spinal fluid had either continued to go down that central canal, the dot that we're drawing on the board right there, or it escaped through those lateral and median apertures into this subarachnoid space and surrounded the brain and spinal cord, giving it that buoyancy. We talked about the choroid plexuses being located in each of those ventricles. They're a bed of capillaries that leak out that cerebral spinal fluid. And you can see they are lined by those ependymal cells or ependymal cells. We can see here those ependymal cells do not produce the cerebral spinal fluid. That's just selective blood plasma that's leaking out of these capillaries. But what they are responsible for is circulating that cerebral spinal fluid with those cilia. They beat in a uniform fashion. They cause this current that allows that cerebral spinal fluid to move in the direction that you see these arrows flowing, from lateral ventricles to the third ventricle, through that cerebral aqueduct down into the fourth ventricle, and from there either into the subarachnoid spaces or into the central canal. And again, at the top, you can see those arachnoid villi, which are the place where this, as you have to think of it, this is kind of a closed system. So as new cerebral spinal fluid is produced, old cerebral spinal fluid has to be drained away at the same pace that new cerebral spinal fluid is produced. Otherwise, you get a buildup of excess cerebral spinal fluid. Or if it drains away faster than it's produced, then you lose that buoyancy, which also, again, causes the problems. So we've seen the choroid plexus. This is a child that has a blockage in these arachnoid villi. So if the cerebral spinal fluid can't escape, then you have a buildup of that cerebral spinal fluid in those subarachnoid spaces. Ventricles, like we said, we put these on the board last time. We found them on the models. There are those two C-shaped lateral ventricles in either cerebral hemisphere. They both drain their fluid into the third ventricle. And then from the third ventricle that is found kind of between the two thalami, We see that fluid goes down to the fourth ventricle. Each ventricle and even that central canal are going to be lined by those ependymal cells. I feel like we covered this last time. Right at the end of our lecture, we can see those lateral ventricles from the side. What kind of looks like a bird beak to me. This is representing the third ventricle. And that little dot in the center there is that interthalamic adhesion. Looking at it directly from the anterior view, there is the third ventricle. We can't see that hole in the side, that interthalamic adhesion from the anterior view, but you can see how that third ventricle connects to the fourth ventricle down here between the cerebellum and the pons. Spinal cord stuff. We started here, and now we're finally getting back to some of the slides that make us think about the spinal cord again. We've drawn the structures of it. But if we want to think about where the spinal cord begins and where it ends, we've got a spinal cord model that is stable right now, but sometimes that falls over. The spinal cord is going to begin at the foramen magnum, that hole in the base of the skull. So we have that occipital bone on this model, and spinal cord starts right there at the foramen magnum, but it's going to go down to about the first or second lumbar vertebrae, and it's going to stop. If we can zoom into the lumbar vertebrae, here we go. So the spinal cord starts right there at the foramen magnum. It goes all the way down to about the first or second lumbar vertebrae, and then the spinal cord stops. What we see beneath that are just a collection of these spinal nerves. It almost looks like the tail of a horse with all the hair of a tail of a horse, and that's what really gives this part of the spinal cord its name. It's called the cauda equina, that just means tail of horse. And if we can zoom into that area, let's see, here we've zoomed in to a part of the lumbar vertebrae where we're just beneath the L1 and L2. So you can see they've labeled these L4 and L5, and this is typically the location where an epidural is given. Okay, so we've got that. If we're thinking about spinal cords, like we said, we know where it stops and where it ends. There is also a distinction between spinal nerve. There should be 31 pair of these spinal nerves, and we know spinal nerves are where the ventral root and the dorsal root merge. They've come together. Spinal nerves are all mixed. They're all two-way streets. They've got sensory and motor pathways. And here's our cross-section through the spinal cord. This one looks just like the model that we have that still has the vertebrae around it. And when there's the vertebrae around it still, you can use the structures that we remembered earlier in the semester. There's that, what am I trying to say? Spinoous process. I almost lost it there. Spinoous process, this also tells us posterior or dorsal side. The body of the vertebrae tells us anterior or ventral side. And sure enough, when we look at the spinal cord, there's our anterior median fissure and our posterior median sulcus. We can see that dorsal root, that always shows us dorsal side. There's the dorsal root ganglion. The ventral root does not have a ganglion, so it kind of helps orient ourselves between dorsal and anterior. The other thing that I like about the model that has the vertebrae around it, like I said, it's the only one that shows those meningeal layers. So we can see that outermost Dura Mater layer. Underneath that Dura Mater is the Arachnoid Mater. And you can still see those little spidery-like projections that are part of that Arachnoid Mater. If we were walking through those little spidery-like projections, we would be in the subarachnoid space. Whoa. So there, that dark area is all the subarachnoid spaces. And then directly touching that spinal cord itself is the pia mater. So we've got those. Then we've labeled our spinal cord already. We've labeled spinal nerve, where the dorsal root and ventral root come together. As we approach the spinal cord, we can see this anterior gray horn and posterior gray horn, lateral gray horn off to the side. You can also see pia mater, arachnoid mater, and Dura Mater. If we added the white columns or white funiculi on there, like we did to our diagram, I think that covers just about all of it, because we've got the white columns, we've got this white commissure, there's the central canal, and kind of hard to see on this image, but better to see here. You can see that gray commissure. which is where the gray tissue from either half of that spinal cord are connected.